get ready to pay more for crabs.
Yeah, and this is not because of inflation or tariffs.
In 2012, a group of scientists did the unthinkable. They took 10,000 lab-grown blue crabs and dumped them into a toxic dead zone where nothing could breathe. Most called it a suicide mission, others called it a waste of money. But what happened next didn't just defy logic, it triggered a biological revolution that is now saving the largest estuary in America. Today, we're diving deep into the secret war beneath the waves to see how an army of 10-legged soldiers is winning a battle that technology alone couldn't. The collapse of the protein factory. To understand why this gamble was so desperate, you have to understand what the Chesapeake Bay used to be. For centuries, this wasn't just a body of water. It was the world's great protein factory. Back in the 1800s, the bay was so thick with oysters that ships would run aground on the reefs. We are talking about 20 million bushels of oysters pulled out every single year. These weren't just snacks, they were the lungs of the bay. A single oyster can filter up to 50 gallons of water a day. Back then, the entire volume of the Chesapeake, nearly 18 trillion gallons of water, was filtered by oysters every 3 to 4 days. The water was gin clear. You could see the bottom 20 feet down. But humans did what we do best. We over-engineered the system for profit. We scraped the reefs bare. By the early 2000s, we had destroyed 99% of the oyster population. And when the filters disappeared, the nightmare began. The Chesapeake is a giant funnel. It drains 64,000 square miles across 6 states, from the industrial farms of Pennsylvania to the suburban lawns of Virginia. Every time it rains, millions of pounds of nitrogen and phosphorus, the superfuel of fertilizer, flow straight into the bay. In the water, this fertilizer does exactly what it does on land. It makes things grow. But instead of corn or barley, it grows algae. Massive, choking blooms that block the sun. When that algae dies, it sinks to the bottom and rots. And here is the chemistry of death. The decomposition process sucks every last molecule of oxygen out of the water. Scientists call these hypoxic zones. We call them dead zones. By 2012, these zones covered nearly a third of the bay. Thousands of square miles where the oxygen level dropped below 2 milligrams per liter. For a fish or a crab, that's like being trapped in a room where the air is slowly being replaced by carbon monoxide. You can't run, you can't hide, and you can't breathe. The Chesapeake was becoming a 4,500 square mile graveyard, the reverse logic of Dr. Anson Hines. In the world of environmental restoration, there is a standard rule book. Rule number 1, clean the water first, then bring back the animals. It makes sense, right? You don't put a canary into a coal mine if you already know the air is poisonous. But Dr. Anson Hines and his team at the Smithsonian Environmental Research Center, (SERC), decided to throw the rulebook into the trash. They asked a radical question: what if the animals are the only ones who can clean the water? They chose the blue crab Calanectes sapidus. Its name literally means beautiful, savory swimmer, but don't let the name fool you. These things are the Hell's Angels of the estuary. They are aggressive, highly mobile, and they are generalist scavengers. Hines' logic was based on the concept of the keystone species. He realized that the bay's ecosystem was stuck in a feedback loop of death. The water was too dirty for animals to live, and because the animals were gone, the waste just kept piling up. To break the loop, he needed a shock force. But he couldn't just use wild crabs. Wild baby crabs have a survival rate of less than 1%. Most are eaten within minutes of hatching. To win this war, he needed super soldiers. At the University of Maryland's IMET facility, they began training these soldiers. These crabs were raised in optimized tanks, fed a high-protein diet, and protected during their most vulnerable larval stages. By the time they were ready for release, they were late juveniles about the size of a silver dollar. They were healthy, they were fast, and most importantly, they were past the stage where everything in the ocean could eat them. The critics were brutal. Many in the scientific community called it ecological theater. They argued that releasing 10,000 crabs into the Lafayette River, a tributary so polluted, it was nicknamed the lung of the term city, was inhumane. They predicted a 100% mortality rate within 48 hours. Dr. Hines ignored the noise. In June 2012, the trucks loaded with oxygenated tanks pulled up to the muddy banks of the Lafayette. It was time to see if 10,000 hungry scavengers could succeed where 40 years of policy had failed. The invisible alchemy of the crab. Why would a crab help a dead river? It's not because they are environmentally conscious. It's because of their unique biological role as the janitors of the benthos. When the 10,000 crabs hit the water, they didn't die. In fact, they thrived. And this is where microbiology gets fascinating. In a dead zone, the bottom is covered in organic sludge. This is basically a thick carpet of dead algae and rotting fish. This sludge fuels anaerobic bacteria that produce hydrogen sulfide, a gas that is toxic to almost everything. But crabs are bioturbators. As soon as they hit the bottom, they began to dig. They dig to hide from predators, they dig to find worms, and they dig just to establish territory. This act of tilling the mud is like a farmer plowing a field. It breaks up the toxic sludge and allows oxygenated water to penetrate the top few inches of the soil. This aeration changes the chemical profile of the riverbed. It allows aerobic bacteria to return. These are the good microbes that break down waste without sucking the oxygen out of the water. But the crabs did something even more direct. They ate the problem. Blue crabs are detritivores. They devour the decaying matter before it can rot and fuel the next algae bloom. Think of them as a massive fleet of underwater vacuum cleaners. Every gram of rotting fish a crab eats is a gram of waste that won't be turned into a death gas by bacteria. Within just 3 months, the sensors in the Lafayette River started picking up a heartbeat. The dissolved oxygen levels at the bottom weren't just stabilizing, they were climbing. For the first time in decades, the riverbed wasn't a graveyard. It was a construction site, and the crabs were just getting started. They were about to trigger a trophic cascade that would bring the giants back to the bay. The trophic cascade, the dominoes fall. In biology, there is a phenomenon called a trophic cascade. Imagine a row of dominoes representing the food chain. If you knock over the one at the top or the one in the middle, the energy flow of the entire system changes. In the Lafayette River, the blue crabs were the middle domino. Before they arrived, the river bottom was a chaotic, unregulated mess. Without a top predator, small, opportunistic fish and polychaete worms were overpopulating. They were stripping the riverbed of every resource, leaving nothing for the larger species. But when the 10,000 blue armada arrived, the rules changed. The crabs began to hunt. They kept the populations of small fish and invasive snails in check. And this created room for the heroes of the Chesapeake to return, the oysters and clams. You see, baby oysters' spat need a clean surface to attach to. If the river bottom is covered in slime and unchecked snails, the oysters can't grow. But the crabs were like a cleaning crew, scraping the surfaces clean. As the crab population stabilized, we saw a sudden, miraculous rebound in wild mollusk populations. And more mollusks meant exponential filtration. If a crab is a vacuum cleaner, a clam is a high-tech water purifier. As the clams returned, the water clarity in the Lafayette River improved by 25% in just 18 months. Clearer water meant that sunlight could finally reach the bottom again. This triggered the return of the holy grail of the bay, submerged aquatic vegetation, SAV, or seagrass. Seagrass is more than just a plant, it's a nursery. It's where baby crabs hide from predators, where fish lay their eggs, and most importantly, it's one of the most powerful carbon sinks on Earth. Seagrass can store carbon 35 times faster than a tropical rainforest. By bringing back the crabs, we weren't just cleaning a river, we were building a massive underwater lung to fight global warming. The ghost in the shell, tagging technology. How do you track 10,000 crabs in a river that looks like chocolate milk? This was the biggest technical challenge for Dr. Hines and his team. Usually, you would just tag an animal. But crabs have a trick that makes tracking them a nightmare. Molting. Every few months, a crab literally crawls out of its own skin to grow a bigger one. If you glue a tag to a crab's shell, that tag will be sitting at the bottom of the river within weeks, while the crab swims away in a brand new, untagged body. To solve this, the Cirque team used microwire tags. These are tiny, hair-thin pieces of stainless steel, etched with a microscopic code. Instead of attaching them to the outside, researchers injected them into the hepatopancreas, or the soft muscle tissue at the base of the swimming leg. When a crab molts, it leaves its shell behind, but it keeps its internal organs in the tag. But they didn't stop there. For the elite soldiers of the group, they used acoustic telemetry. They attached high-frequency pingers to a small subset of the crabs. These pingers sent out a unique ID signal every 60 seconds. The team placed a grid of underwater microphones, hydrophones, across the entire Lafayette River. For the first time in history, we could watch the war for the river in real time. We saw that the lab-grown crabs weren't just surviving, they were out-competing the wild ones. They were faster, they were more aggressive, and they were establishing vanguard territories in the most polluted parts of the river. This data proved that active intervention worked. It wasn't just a drop in the ocean, it was a permanent change in the river's DNA. The return of the giants. By 2015, something even more incredible happened. The crab signal had reached the Atlantic Ocean. When an ecosystem begins to heal, the news spreads through the water like a chemical broadcast. The return of the blue crabs and the rebounding fish populations meant that the buffet was open for business. Researchers began to spot Kauno's rays, massive, wing-like creatures migrating deep into the Lafayette River. Before the crabs, these rays stayed away because there wasn't enough high-protein food to justify the trip. Now, they were back, hunting the surplus of crabs and keeping the population balanced. Then came the dolphins. Bottlenose dolphins started appearing in the upper reaches of the bay, in numbers not seen since the 1960s. They weren't just passing through, they were staycationing. They were following the massive schools of menhaden and croaker that were now thriving in the oxygen-rich, grass-filled waters. And finally, the ultimate sign of success, the apex predators. Small sharks, including sandbar and bull sharks, were caught and tagged in the restored zones. In biology, the presence of a top-tier predator is the gold medal of restoration. It means the entire food web, from the microscopic bacteria in the mud to the 200-pound shark at the top, is functioning again. The dead zone was no longer a graveyard. It had become a high-energy, high-traffic biological highway. The gamble of 2012 had paid off. But as the program prepared to go bay-wide, a new controversy emerged: the genetic dilemma. If we flood the entire bay with lab-grown crabs, are we destroying the wildness of the Chesapeake forever? The genetic dilemma, clones vs. survivors. As the Blue Armada expanded from 10,000 to over 500,000 crabs by 2020, a new, quieter crisis began to worry the world's top geneticists. It's a problem of DNA homogeneity. When you raise 500,000 crabs in a hatchery, they often come from a very small pool of broodstock, the mothers and fathers. If all the crabs we release are siblings or cousins, we risk creating a genetic monoculture. In the wild, diversity is life. If a new virus or a sudden spike in water temperature hits the bay, a diverse population will have some individuals with the magic gene to survive. But if they are all genetically identical, a single disease could wipe out the entire restoration project in a week. To solve this, the University of Maryland developed a wild infusion protocol. Every season, they capture sponge crabs' wild females carrying millions of eggs from different parts of the bay. They hatch the eggs in the lab, raise them to super soldier size, and then release them back into a different part of the bay. This genetic mixing ensures that the Chesapeake crab population stays as wild as possible, even if they spent their childhood in a plastic tank. It's a high-tech game of musical chairs. By moving genes from the north of the bay to the south, we are actually accelerating evolution. We are helping the crabs adapt to the polluted zones faster than they ever could on their own. We aren't just restoring a population, we are patching their software for a tougher, warmer world. The waterman's redemption. But science is only half the story. To truly save the Chesapeake, you have to win the hearts of the watermen. These are the men and women whose families have lived on the bay for 300 years. To them, the blue crab isn't a data point or a bioturbator. It's their mortgage, their children's tuition, and their entire identity. When the population collapsed by 70%, the culture of the bay almost died with it. Towns like Crisfield and Smith Island were becoming ghost towns. At first, the watermen were the biggest critics of the lab release program. They saw it as playing God. They feared that lab-grown crabs would be weak or tasteless. But then, they started catching them. The tags proved it. The hatchery crabs were bigger, meatier, and more aggressive than the struggling wild ones. The watermen saw their harvest stabilize. They saw the dead zones, places where they hadn't dropped a pot in 20 years, suddenly teeming with life. By 2024, the relationship changed from conflict to collaboration. Today, many watermen act as citizen scientists. They report tagged crabs, They help transport juveniles, and they have become the fiercest protectors of the seagrass beds. They realized that the old bay was gone, but a new bay was possible. It's a bay where industry and nature don't have to be enemies. One veteran waterman put it best. We used to think we were taking from the bay. Now we realize we're part of it. If the crab doesn't breathe, we don't eat. Beyond the bay, the blueprint for the planet. The success of the Lafayette experiment has sent shockwaves far beyond the Atlantic coast. The lesson we learned that you can jumpstart a dead system by injecting Keystone engineers is now being applied to the world's most fragile ecosystems. In the Gulf of Mexico, they are using a similar super soldier approach with Caribbean king crabs. These crabs are the lawnmowers of the coral reef. When the reefs get choked by algae due to fertilizer runoff, the corals die. By releasing lab-grown king crabs, we are seeing reefs breathe again in months, not decades. In California, they are looking at sea otter reintroduction to save the kelp forests. Just like the blue crab, the otter is the general that controls the system. Without them, sea urchins eat all the kelp. With them, the forest returns, storing massive amounts of carbon. We are entering the era of biological engineering. For 50 years, we tried to fix the environment with hardware, bigger pipes, better filters, more concrete. It was expensive and it often failed. Now we are switching to software. We are realizing that a crab, a beaver, or an oyster is a self-replicating, self-repairing, solar-powered machine that has been programmed by 400 million years of evolution to fix exactly the problems we are causing. The Chesapeake formula is simple. Identify the engineer, strengthen the engineer, get out of the way. But as we look toward the 2030s, we have to face the ultimate question. If we can engineer nature back to life, does that give us a free pass to keep polluting? Or are we just buying ourselves a little more time before the final collapse? The 2030 vision, the Blue Revolution. As we look toward the year 2030, the Chesapeake experiment has evolved into a global blueprint for the Blue Revolution. Imagine a future where the phrase dead zone is a relic of history books. By 2030, the program aims to have released over 10 million juvenile crabs into every major tributary of the bay. But the goal isn't just numbers, it's autonomy. The ultimate sign of success in restoration is when you don't have to do anything anymore. When the crabs mate in the restored grass beds, when their larvae survive because the water is clean, and when the oysters filter the water because the crabs protect them, that is a self-sustaining system. By 2030, the Chesapeake Bay is projected to reach its healthiest state in 150 years. We are seeing the return of the sturgeon, a prehistoric fish that hadn't spawned in these waters for decades. We are seeing the carbon sequestration of the bay's seagrass meadows, offsetting the emissions of entire cities like Baltimore and Norfolk. But this success brings us to a dangerous crossroad. The moral hazard. If we can just release more crabs to fix a polluted river, does that give us a license to keep dumping fertilizer? If we can engineer nature to survive our waste, do we lose the motivation to stop wasting? This is the central tension of the 21st century. The blue crabs are not a get-out-of-jail-free card for industrial farming. They are an emergency life support system. They are buying us time-valuable years to fix our drainage systems, transition to regenerative agriculture, and rethink how we manage the land. The crabs are doing their job. Now it's time for humans to do ours. The power of the repair crew. So, did 10,000 crabs really save the Chesapeake Bay? The answer is, they started a fire. They were the spark that proved life is more resilient than we ever imagined. They proved that nature isn't just a victim of our progress. It's a powerful ally waiting for an invitation to fight back. We spent 50 years treating the bay like a broken machine. We tried to fix it with chemistry and hardware. We failed. But when we treated it like a living organism, when we focused on its immune system, the crabs, the how, and the grass, the bay began to heal itself. This is a lesson for every crisis we face. Whether it's the disappearing forests of Southeast Asia, the dying reefs of the Pacific, or the warming rivers of Europe, the repair crew is already there. They are submerged in the muck, they are hidden in the soil, and they are waiting for us to stop destroying their tools. The Blue Armada of the Lafayette River isn't just a story about crabs, it's a story about humility. It's about humans admitting that a 10-legged scavenger with a brain the size of a pea is a better engineer than our most advanced computers. We are moving from the era of conquest to the era of collaboration. And in this new world, the heroes don't always wear capes. Sometimes they have blue claws, they love to eat dead fish, and they're not afraid to snap at your fingers if you get too close. The power of the small. A single crab doesn't clean a bay, but 10,000 crabs create a sanctuary, and 1,000,000 crabs create a future. What is the blue crab in your community? Is it a local wetland that needs protection? Is it a native plant that needs to be returned to your garden? Is it a voice that needs to speak up for a dying stream? We're the ones who broke the system, but we are also the only ones who can give nature the fighting chance it needs to fix it. The crabs have shown us the way. They've done the hard work of digging through the toxic mud. Now it's our turn to keep the water clear. Thanks for joining us for this deep dive. If you believe that nature is the best technology we have, hit that like button. Share this story with someone who thinks the world is beyond saving. And don't forget to subscribe to The Ultimate Reveal for more stories about the incredible architects of our planet.